Welcome. Today I am going to discuss about the salient features, vegetative and sexual reproduction, spore dispersal mechanism in Marchantiales. The Marchantiales belonging to the division Marchantiophyta, class Marchantiopsida, subclass Marchantidae and order Marchantiales. This is an order of Bryophyta consists of thyroid species and plants are not divisible into leaf, stem and rhizoids. It includes 25 genera and 400 species. The Smith has divided Mercantiales into 5 families. They are Rexiaceae, Coccineaceae, Targionaceae, Monoclyaceae, Mercantiaceae. As in other bryophytes, the gametophyte generation is dominant, with the sporophyte acting as a short-lived part of the life cycle, dependent upon the gametophyte. The gametophyte of Mercantiales are prostrate, ribbon-shaped, dichotomously branched and dosiventrally differentiated. This is a Rexia thallus showing rosette form. The general characters of Mercantiales. The plant body is gametophyte, which is usually a prostrate, dorsiventrally dichotomously branched thallus with a more or less marked midrib. Thallus often of complex anatomy with air pores on the dorsal surface. Air chambers with chlorophyll cells are forming a photosynthetic area, and the cells of the remaining are serving as for storage. The genus of liverworts in the order Mercantiales commonly found on moist clay sickly soils, especially on recently burned land throughout the northern hemisphere. The gametophyte of Mercantiales are ribbon-shaped, dichotomously branched, and dosiventrally differentiated. Internal tissue are differentiated into two distinct regions, the dorsal and ventral region. The dorsal region is green, is a similatory zone which is generally enclosed air spaces, also called air chambers. The air chambers communicate with the exterior generally through pores. The ventral region is composed of compact colorless parenchymatous storage tissue. Scales are usually present on the ventral surface of the thallus. The ventral surface of the thallus bears scales and two kinds of rhizoid, smooth-walled and tuberculate. The thallus are attached to the substratum generally by means of rhizoids. Male and female plants have umbrella-like stalked reproductive structure. The male structures are disc-shaped with scalloped edges. The female structures have 9 finger-like projections. The sporophyte develop from fertilized eggs in the female structure. The gametophytes of Mercantius differ from those other Hepaticeae in that they are internally differentiated into various tissues. The structure and development of the sex organs differ from that of all others. The sporophyte are also distinct from others. The jacket layer surrounding the sporogenous tissue is but one cell in thickness. The sporogonium is simple in structure and small, either with or without seta. It has a capsule wall on one cell thickness. Columella is absent. A capsule dices by various ways but by never by 4 regular valves. The structure of gametophyte in Mercantiales. The gametophyte is the main plant body. Its external morphology is different in different classes. In Monoclyaceae, the simplest thallus is observed, which are dichotomously branched, midrib also branched. Monoclea forestry is the largest thallus of bryophyte at the dorsal region involucral present in which sex organs occur. In Rexiaceae, a small thallus unbranched midrib is present. Plants occur in colonies, mature rhizoids like protoplasm. In Coccineaceae, thallus are long 2 to 3 times dichotomously branched and ribbon shaped. Midrib is also present and it is branched. In Targionaceae, the shape of the thallus is uneven adventitious. Branched tubers also occur. In Mercantiaceae, the thallus is large and 2 to 3 times dichotomously branched. Gamma cup is present in the midrib at a dorsal portion. Mercantiace's reproduction is by vegetative, asexual and sexual methods. Vegetative reproduction occurs in both male and female plants by means of rinded, fringed gammae or by fragmentation. Asexual reproduction in Mercantiales by fragmentation of forming specialized structures known as gammae, which are asexual but they are formed on the receptacles known as gamma cups. Gamma cups are present on the dorsal surface near the midrib of the gametophytic thalli. Gamma are multicellular and green. Sexual reproductive structures of Mercantiales. In Rexiaceae, the sexual reproductive structures are inserted in the thallus, while in Coccineaceae, Sex organs are present on the receptacle at the dorsal surface. In Targionaceae, sex organs are present at the lateral branches, which arises from the ventral surface. In Monocliniaceae, sex organs are present in the sessile receptacle, which are present at the dorsal surface. While in Marchantiales, specific organs are formed for sex organs, named as antheridium and archegonium. Antheridium is a male sex organ which is found on the male thallus, which is developed from the antheridial initial cell. The antheridial cell now divides transverse division and longitudinal division form four-celled structure, while the stalk cell is divided periclinal division to form the jacket initial and central cell. The jacket initially continues to divide from the jacket divide to form the anthrozoid and cells and mother cell. The anthrozoid mother cell forms the anthrozoid which is biflagellated and helical. When the anthrozoid mature, anthrozoid mother cell rupture to release the anthrozoid. Next is archegonium. Archegonium is the female sex organ developed from the female thallus. The basal cell divides to form the stalk, while the upper cell is divided to form two longitudinal division to form peripheral and central cell. Peripheral cell divides and transfer division to form primary cover cell and primary jacket cell. The primary cell divides to form the jacket. The spore dispersal mechanism is by a spore generating structure called a capsule forms at the top of the sporophyte. Spores are generated by meiosis, and as capsules mature it dries and finally pops open, releasing the spores for dispersal. Given appropriate conditions, the spores will develop into new gametophyte. For fertilization, when archegonium matures, the anthrozoid is released towards the archegonium. Neck canal cells and the ventral canal cells dissolved and water enters in the neck portion. From the neck of the archegonium, some salts and proteins are excreted which simulate the anthrozoid to move in that direction. Then the anthrozoids enter the venter by only one of them fertilizes with the egg and forming a diploid zygote. The development of sporophyte in Marchantiales. Immediately after formation, the zygote increases in size. The zygote is divided by transverse division to form epibasal and hypobasal cell. Hypobasal cell form foot, epibasal cell contribute to form the capsule. In Coccineaceae, only the foot and capsule are formed, and in the capsule spore is tetrad is present. In Targionaceae, the sporophyte is divided into foot, seta and capsule. In Monocliniaceae and Marchantiaceae, well-developed foot, seta, capsule are present, and spore tetrad is present. This is all about my topic. Thank you.